Tiny shellfish, massive profits, and cleaner oceans. Welcome to the fascinating world of mollusc culture. Aquaculture has evolved beyond fish farming, and mollusc culture now represents a sustainable intersection of food security, environmental management, and coastal economics. In this video, you will learn about mollusc culture. After watching this video, you will be able to understand mollusc culture system, spat collection, spat collector, hatchery production of spat, fecundity rate, grow-out methods, hanging method, bottom culture method, factors affecting grow-out, harvesting and pros and cons of scallop farming. Scallop are marine bivalve mollusk. Scallop, marine bivalve mollusk. Scallops are members of the mollusc class Bivalvia that also includes clams, oysters, and mussels. As with most bivalves, hence the name Bivalvia, two valves. Scallops have two-part calcareous shells, or valves, that are hinged and more or less symmetrical, whereas a mollusk typically has a body divided into the three parts of head, visceral mass, and muscular foot. Bivalves lack a distinct head and head structures are much reduced. Unlike most bivalves, but like adult oysters and giant clams, scallops lack a foot. Marine bivalve mollusks belong to family Pectinidae. This is the largest family of living bivalve. They have more than 300 species. They have considerable importance in the seafood industry. Its commercial scale culture is limited to Japan and China. Scallop is characterized by a large, well-developed central adductor muscle. Muscular foot absent. Two valves typically being fan-shaped, often with a radiating fluted pattern, and with flared wing-like projections on both sides of the straight hinge. Mostly free-living. Some species can attach to a substrate by a structure called a byssus. A byssus is a bundle of filaments secreted by many species of bivalve mollusk that function to attach the mollusk to a solid surface. Species from several families of clams have a byssus, including pen shells, true mussels, and Dreissenidae. A free-living scallop can swim by rapidly opening and closing its shell. This method of locomotion is also a defense technique, protecting it from threatening predators. Scallops feed by filtering microscopic animals and plants from the water via cilia. The scallop are dioecious. Males and females are separate. Some are hermaphrodites, both sexes in the same individual. A few are protoandrous hermaphrodites, males when young then switching to female. Spermatozoa and ova are released freely into the water during mating season and fertilized ova sink to the bottom. After several weeks, the immature scallop hatches and the larvae drift in the plankton until settling to the bottom again to grow, usually attaching by means of byssal threads. Some scallops, such as the Atlantic-based scallop Argopecten irradians, are short-lived, while others can live 20 years or more. Culture systems similar to those used for culture of other bivalves. Early efforts involved the collection of seed scallops and their planting in either depleted beds or in areas suited for rapid growth. This led to the next stage of rearing the seed in cages or ponds and releasing them in natural beds after they had grown to a shell length of about 3 mm to improve survival rates. The present trend is to raise the seed to market size using suspended systems. In China, scallops are often cultured in association with seaweed in foreshore areas, in lantern or pearl nets suspended from long lines. To get its seed from ocean spat collectors are used. Spat, juvenile stage of bivalve development, and is perhaps the most commonly used term applied to juveniles in hatcheries. It relates to bivalve larvae that have set and undergone metamorphosis. A variety of substrates have been used for collecting seed scallops. Traditional spat collectors used in Japan are cedar leaves, scallop shells, polyethylene, mesh bags attached to long line systems. This figure showed different types of spat collectors like Shadime sheet, plastic strip, etc. Any substrate with a clean surface can be used for collecting seed of the European scallop C. maximus and C. percularis. The collectors are laid out at times of spatfall, which are forecast on the basis of close monitoring of water temperature, spawning and larval abundance. Length of long lines can be set up to 50 to 100 meters. Number of bags attached to each line depends on the water depth and the number of branch lines. The bags are filled with artificial substrates like mesh monofilament, soft nylon, nylon and plastic mesh, rubberized fiber. Mesh size of these bags depends on the local conditions, most important of which is the silt load in the area. Bag's mesh size must be in the way that it facilitates free water flow, prevent the escape of the settled spat, prevent clogging of the meshes by silt. Hatchery production of spat. Thermal shock, ultraviolet rays, hydrogen peroxide in very low concentrations can be used as stimulus to induce spawning in scallops. Mature scallops can be induced to spawn by thermal shock, and larvae are grown in hatcheries by techniques similar to those for other bivalves. Sexually mature adults can be induced to spawn by increases in temperature and by the addition of milt from mature males in spawning tanks. On the other hand, most of the seed used in culture of C. ferreri and C. nobilis in China are reported to be hatchery produced. The bay scallop, A. irradians, the Chilean scallop, A. purpuratus, the deep sea scallop, P. yesoensis, and the Chinese scallops mentioned above have all been spawned in hatcheries by using thermal shock. Fecundity rate of scallop is high. A fully mature specimen can produce at least 100 million eggs. In spawning hermaphrodite individuals, care has to be taken to avoid polyspermy by collecting the sperm and eggs in separate containers and then artificially fertilizing the eggs. Fertilization. 1 liter of egg suspension can be fertilized by 2 to 6 ml sperm suspension. Embryonic development is rapid and free-swimming trochophores can be obtained in 8 to 24 hours depending on water temperature. The optimum temperature for the development of the scallop is reported to be 26-28 degrees C, and gentle aeration and frequent water exchange are usually maintained in the tanks. The larvae are fed on phytoplanktonic organisms such as Monochrysis, Chaetoceros, Isochrysis, Phaeodactylum, Dunaliella and Tetraselmis. The larvae normally settle within 2 weeks. This is the life cycle of scallop. Adult scallop is less than 18 cm in size. Eggs fertilized externally. Hatching occurs after 24 hours and trochophore larvae emerge from fertilized eggs, which changes to straight hinge larvae within 3 days. After 20 to 30 days, the straight hinge larvae will be changed to pedivliger larvae that converted to newly settled juvenile within 5 to 15 days. The size of this juvenile is approximately 1 millimeter. This juvenile grows to early juvenile within 4 months, which grows to adult within 3 to 4 years. The most important factors that affect the growth rate of scallop include substrate, depth of water, salinity, temperature, exposure to air, wind and water currents, food availability, water quality. Site selection for cultivating scallop is also extremely important. The simplest system of grow-out is by planting in suitable beds. The spat are released from boats during the summer months. The density of spat depends on local conditions, but 5 or 6 per m2 is considered suitable for obtaining a commercial size of 5 or 6 per kg. The survival rate is only about 25-30%. Even in hanging culture, more widely practiced now, the mortality rates are high. Various grow-out methods are used for cultivation of scallops. In this video, we will study all methods one by one. First one is hanging method. raft or long line system that floats on the sea surface from which the cultured scallops are suspended, which include use of pearl nets, lantern, pocket nets, ear hanging, rope culture, plastic trays. Secondly, bottom culture method can be employed. It employs the use of scallop-filled plastic trays or wild ranching. This figure shows the use various grow-out methods. Long lines can be used in shallow inshore areas as well as offshore deep areas. Rafts are. This figure shows the use various grow-out methods. Long lines can be used in shallow inshore areas as well as offshore deep areas. Rafts are only suitable for protected areas. Using such large long line systems, as many as 500,000 scallops are reported to be cultured in an area of 9 ha in sites exposed to strong wave action and wind during winter months. In China, lantern nets suspended on long lines is the main form of culture for all scallops. In bottom culture, seed scallop are planted in plots in the intertidal or shallow subtidal zones. Predator exclusion devices may be used. Lantern method. Spat with hardened shells are placed in lantern nets, known also as adnon baskets, and hung from the long line system. The lantern nets are made of monofilament netting supported by plastic-coated or galvanized wire hoops which divide the net into several compartments. Each compartment has a lateral opening through which scallops can be introduced or removed. The stocking density determines the growth rate, and a density of 20 scallops per compartment is reported to give good results. These nets are suspended by nylon ropes. Pearl net. Pearl nets hung from the long lines are conical or pyramidal in shape, made of small mesh, 2 to 7 mm, net, and provide adequate protection to the spat from predators and silt deposition. Free circulation of water is ensured. The maximum number of spat in each net is about 100 and the nets are hung at 2 to 12 m depth of water. As the growth rate of the spat is rather rapid, the stock has to be thinned out at regular intervals and transferred to new nets. Next to its use of ear hanging, the traditional Japanese method of grow out to market size consists of hanging seed scallops on long lines, sometimes referred to as the ear hanging method. Small holes are drilled on the anterior side of the ear of each seed scallop, about 2 mm in size, threaded with nylon and tied to ropes for hanging on long lines. It normally takes 2 to 3 years for the seed scallops to grow to market size, which for the deep sea scallop in Japan is 10 to 11 cm in length. Scallops are most commonly harvested using scallop dredges or bottom trawls. Scallops are hand-caught on the ocean floor, as opposed to being dragged across the sea floor by a dredge, which causes them to collect sand. In addition, the normal harvesting methods can cause delays of up to 2 weeks before they arrive at market, which can cause the flesh to break down and result in a much shorter shelf life. Times conditions suitable for mollusc culture include depth. Depth of water should be 4 to 10 meters, salinity must be in the range of 32-34 per thousand, temperature 4 to 8 degrees Celsius, lifespan of mollusk is 10 to 12 years. Pros of mollusk farming system include that scallops don't need to have food supplied to them, they clean the water as they filter feed. There aren't any diseases or parasites associated with scallops. Many of the nets are collapsible and easy to store. The cons of mollusk farming system include that this system is labor-intensive. It may cause visual pollution and eutrophication associated with scallop farming. Only the adductor mussel, sometimes roe, is harvested. Availability of reliable seed supply may change with season and climate change. Thanks for watching. Like and share this video. Subscribe this channel to get more informative videos.